Shall I start?
We'll give folks maybe 2 or 3 minutes to join, and then we'll start from there.
Yeah, until we open up the webinar, we can't see how many people are actually present or waiting. Isn't that right, Kavya?
Yeah, so we're live now.
There we go. All right. I see Vito made it on. That's good. We have a number. Got up to 10 at least. That's a good start. Hopefully we'll get a few more here. We'll wait just another couple minutes. Just to remind everyone, this is the Quantitative Systems Biology seminar series. And we are going to be starting in just a couple minutes till we try to get as many people in as we can. It's a different start time for us this morning, 9:30 a.m. So usually we're starting afternoon, but hopefully we'll continue to have more people arrive. So I'll go ahead and start introducing our speaker today, Dr. Seema Mishra from Hyderabad in India. So I've looked into some of her work. I realized she also had spent some time with Andrea Califano, part of one of the lead investigators of one of the centers in the U54 consortium that we are part of as well. And she's done some amazing work on computational analyses in all sorts of fields, including a lot of the COVID pandemic. But today she's going to be talking about systems biology approaches to interrogating roles of long non-coding RNAs in pan-cancer gene expression regulation. So thank you very much for being here today, and I'll let you get started.
Okay. So thank you. Thank you everyone for being here with me today to attend my talk. And thank to Vito and Lois especially. It was a very good coincidence that we met. Actually with Andrea, I didn't know Vito might have heard about me. So it was a happy coincidence that we met there. And now the title of the talk is Network Connection is Instability, saying, is it clear now? Okay. So I'm happy and honored to be with you today. And the title of my talk is again Systems Biology Approaches to Interrogate Roles of Long Non-Coding RNA in Pan-Cancer Gene Expression Regulation. So cancer, as you know, is a proliferative disease. And there are certain hallmarks of cancer, like sustained proliferative signaling, avoiding growth suppressor, activating invasion and metastasis, enabling replicative immortality, inducing angiogenesis and resisting cell death, as we all know much about that. There are certain emerging hallmarks also, such as deregulating cellular energetics, avoiding immune destruction, tumor promoting inflammation, genome instability, and mutation characteristics. Now, these hallmark capabilities arise due to changes in gene expression in cancer, which either activate or inhibit these hallmarks. So, on our way towards understanding cancers, we saw that there is a data set called the Cancer Genome Atlas, which is a high quality, high throughput database of pan-cancer gene expression study. Earlier, from 2008 to 2012 or so, the cancers were analyzed at individual cancer type level. For example, this paper just mentioned glioblastoma or ovarian carcinoma or human colon and rectal cancer because these mostly utilize microarrays. And with the advent of newer RNA-seq technologies, from 2018 onward, the focus shifted to pan-cancer levels of genetic analysis. Now, towards our understanding cancer, if these hallmarks are same or similar across all cancer types, despite their inherent heterogeneity, if yes, can we harness this similar hallmark level to find any common regulatory pattern and to identify cancer drug targets? Because an effective and universal pan-cancer biomarker drug target identification is the need of the hour now. So newer technologies and fields such as RNAs and computational systems biology are helping us drastically changing the understanding of cancer biology. Gene expression changes can be quantified and detected in a highly sensitive and accurate way using RNAs as compared to microarrays, and they also result in novel transcripts discovery. So, using the cancer genome atlas dataset from Illumina high-sec RNSec version 2, we found the gene expression values across 5,600 tumor samples and corresponding normal 601 samples from 15 different types of cancer. There are at least a total of 20,531 genes and their expression values in each sample, and tumor type studies are mentioned here. We first applied unpaired student data and lemma to analyze significantly differentially expressed genes with p-value less than 0.01 and fold change greater than 2. And for immunohistochemistry data, to understand the genes at protein level of expression, we used human protein atlas. Then for functional annotation of identified key genes, we used gene code 3, which is which annotate a gene set on the basis of KGG pathways, gene ontology pathways, and others. So this is the flowchart, the same method as mentioned before. From our analysis, we use t-test and also lemma values because we wanted to eliminate false positives and increase the accuracy of our genes identified. T-test identified 368 common, significant, and differentially expressed genes from between tumors and normals, while lemma found 104 such genes. And when we compared t-test and lemma using Venn diagram, there were 70 common genes between these two, leading to a higher accuracy of our prediction. So these 70 common genes were used further in order to generate a heat map to determine which of these genes are upregulated and which are downregulated genes. So this heat map is for read one type of cancer, the rectal adenocarcinoma data. And these columns are for samples of tumor, and this is for normal sample. And the rows correspond to different genes. We find that there is a clear differential expression. Refrigerant squares corresponding to lower expressions and blue squares corresponding to red correspond to higher expression and blue squares correspond to lower expressions of the genes. So we see that these genes are more expressed in tumors as compared to normal, while some of these genes are less expressed in tumors as compared to normal sample. So this pattern of up and down regulation is maintained across all the tumor types there, from PLCA to BRCA to KIRP and so on. So also, because these genes have been analyzed at mRNA level, we need to analyze it at protein level also, because there is disparity in the abundance of mRNA and protein inside a cell. If there is a high level of mRNA, there should be a corresponding higher level of protein, but this is not always so. So we wanted to study a protein expression level of these genes identified using immunohistochemistry intensity data from human protein atlas. This intensity data ranges from not detected to low to medium to high level, with darker blue corresponding to higher level and white corresponding to undetected level. Now, from this, these are representative cases for FOXM1 and ORCA genes. In for example, case, mRNA levels are upregulated in cancer, and they are also seen to be upregulated at protein levels across cancer types. In our case, mRNA levels are upregulated in cancer, but at protein levels, things are downregulated. So we say that at the level of FOXM1, things are positively correlated genes, and at the level of ORCA, things are negatively correlated genes. So likewise, we got some genes from our sets that are positively correlated with similar expression levels of both mRNA and protein. And there are some genes that are negatively correlated as well. Other genes we were able to establish clear correlation. So we wanted to use those genes that have positive correlation with protein expression levels, and these genes have been studied further. So this is annotation analysis using gene code 3 to understand what functional types the positively correlated genes correspond to. This table shows that most of the positively correlated genes correspond to DNA-dependent regulation of transcription biological processes, as well as gene expression, signal transduction, cell cycle, DNA repair, mitotic cell cycle, and cell proliferation process. So here, while it is intuitive that most of the genes may be involved in cell cycle or transcription regulation, it is also of interest to note that there are DNA repair proteins also, proteins involved in DNA repair. Because if there is a apparent DNA repair, then there will be mutations and there will be the occurrence of cancer. So the conclusion of the study is that choosing RNA a divergent set of genes apart from the usual set, which is generally P53, Mike et al., is identified as a common thread running across all cancers. This means that usually there are P53, there is a Mike, and there are other genes that are well known. But we found some other genes also that are not known so much. So we further wanted to know which molecule or a molecular fashion is the master regulator of these genes. Now everyone knows that proteins are usually master regulators, but we wanted to find out if there are some novel regulators as well. We zeroed in on long non-coding RNAs because they have been apparently expressed in cancer, and studies have shown that long non-coding RNAs may also act as tumor suppressors or oncogenes. Now the number of LNC-RNA genes in a human cell outnumber protein coding genes, and these number about 60,000. And they are mostly regulatory in nature, regulating complex cellular behavior by epigenetic regulation. Okay, so cancer-related LNC-RNA are typically found by copy number alterations or genomic sequences or changes in gene expression. And cell culture and animal model functional study link these LNC-RNA to the cancer phenotypes. See, all these phenotypes are the hallmarks of cancer. LNC-RNA mode of actions is through chromatin modifying complexes. They modify chromatin and act as a transcriptional regulator activity. They also function as an enhancer RNA in the nucleus. This occurs in the nucleus. And in cytoplasm, these affect the mRNA stability or mRNA translation and thus affect mRNA expression level. These are also microRNA competitors, and some LNC RNA are also known to code small peptides called small ORFs. Now for our study, we used LNC RNA database, which harbor significant differentially expressed LNC RNA gene in several cancer types taken from TCGN datasets. And we also wanted to understand the interaction of LNC RNA with mRNA. And we used this human LNC RNA interaction database hosted by Japan. For our integrated network analysis, we used ORAT database. So the flowchart is more or less the same as protein coding genes. LNC-RNA were also analyzed using differential expression followed by heat map analysis, followed by functional enrichment analysis, with regulatory network constriction and prediction of LNC-RNA-mRNA interactions and so on. So we found differential expressed LNC-RNA 24 differential expressed LNC-RNA in 9 types of cancer common to those cancer types which were taken in the coding genes expression analysis. And upregulated LNC-RNA out of things for PVT1, UHRF1, SNHG11, and so on. And there were 2 down-regulated LNC-RNA in tumor called MAG2 and MYR22HG. Rest of the LNC-RNA were tumor-specific differentially expressed LNC-RNA. Since we wanted to take those LNC-RNA which have a common pattern of expression across all the tumor types, only things upregulated and downregulated LNC-RNA across all the tumor types were taken further into consideration. So mapping the cancer hallmarks and the genes, we found that most of the genes are involved in sustaining proliferative signaling, along with all of the differential expressed LNC-RNA. Another gene set is involved in enabling replicative immortality. Both coding genes and all the differential expressed LNC-RNA study. And the differential expressed LNC-RNA are also involved in inducing angiogenesis and in resisting cell death. We could not find any awaiting growth suppressor, any genes involved in awaiting growth suppressor or activating invasion and metastasis because the dataset we analyzed was primary tumor dataset and not secondary tumors or metastatic tumors. Now this is the table for the interaction prediction of selected coding and non-coding genes. We see that this LNC-RNA AC00554.5 is interacting with these protein coding genes at 5' QTR or CDS or 3' QTR region. And from this table, we came to know that while multiple LNC-RNA such as this one and MAG2 are interacting with CAT2B and SAN1 and PCGF5, the same gene can be interacting with multiple molecules. So this is a multiple LNC-RNA to multiple genes or multiple gene to multiple LNC-RNA interaction process. So the conclusions of this study is that LNC-RNA fraction with members such as PVT1, SNHG11, QHRF1 can perform its role as the master regulator of the gene through direct or indirect interaction. Now we wanted to find out how exactly are these genes interacting in a signaling or regulatory network? How are these regulating each other? So we perform network analysis of the significantly differentially expressed coding genes and non-coding genes. And this is the network analyzed from cytoscape. And the circles are the coding genes. Red circles correspond to upregulated genes. Green circles correspond to downregulated genes. While triangles are for LNC-RNA genes, with magenta colored triangle for upregulated and blue colored triangle for downregulated LNC-RNA. So this network has a total of 130 nodes and 311 genomes. Further, we characterize this network on the basis of certain quantitative parameters such as degree, high clustering coefficient, or betweenness centrality. Degree just means tendency of a node to be connected to other node, while clustering coefficient means these genes cluster together as a tight clustering unit, and betweenness centrality means number of common paths going through them. So we also analyze this network on the basis of bottleneck nodes, such as hub bottleneck, non-hub bottleneck, hub non-bottleneck node, or non-hub non-bottleneck nodes. So we found that E2F1 has the highest in degree, while E2F2 has the highest clustering coefficient. E2F1 also has the highest betweenness centrality. While from LNC-RNA dataset, PVT1 has the highest in degree. So these were taken for further analysis from this is some network of the network shown before, we find that PVT1 is connected to ETS1, NOP2 and to FOXM1. So NOP2 and FOXM1 are on our list of differentially expressed coding genes. And these interactions were taken from literature research, as pneumonia, which was used to construct this network, did not specify it. So we see from this that PMT transcription is regulated by FOXM1. PMT1 also directly binds and stabilizes FOXM1 and NOP2 proteins in a feed forward or feedback network. So this is the subnetwork of E2F1. We see that E2F1 is also directly connected with FOXM1 and which is connected with PMT1 here. So we zeroed in on the genes, key coding genes and LNC-RNA genes interacting with each other as a common thread running across all cancer types are E2F1, ETS1, PMT1 and FOXM1. So there is a novel gene called SNHG11, which is a novel LNC-RNA identified from our study. While function of SNHG11 is unknown, we hypothesize that primarily bird expression, interaction, and network patterns similar to PBT1, its function can be predicted by direct extrapolation. So SNHG11 we need to study this further. So we have derived a unified model of common coding genes regulation. In this E2F1 transcription factor upregulates FOXM1 transcription, and while FOXM1 upregulates PVT1 transcription. PVT1 after being transcribed binds to other coding genes at 5' UTR or CDS or 3' UTR region and may influence these genes at the level of mRNA splicing or mRNA stability process. So we find that E2F1, FOXM1 and PMT1 form a regulatory axis which was identified as a distinct pan-cancer regulatory pattern across 15 different cancer types. We also further characterize structural basis of the LNCRNA-mRNA interactions by predicting LNCRNA structure. Now we see that not much LNC-RNA structures are available in PDB because its structure is difficult to decipher. But there are some LNC-RNA structures available, experimental ones such as HOTAS and SRA. So this is the experimentally available hot air structure, and this is the RNAFOLD structure, computational structure. We found that the computational structures correspond to the experimental interactive structure, leading to a higher accuracy of RNAFOLD prediction. In this nucleotide 1 to 513 domain consists of 12 helices, 8 terminal loops, and 4 junctions. Computational interactive structure also on the same 1 to 513 nucleotide consists of 14 helices, which is near to 12 helices and 4 junctions. And so we have more confidence in RNAFOLD predictions now. So we have derived PMT1 structure using RNAFOLD, DRN-J9, TRMT1 and RNFP1 structure using RNAFOLD, and these are other gene structures now. So we found the number of secondary structures in each of the genes into helix, hairpin loop, Pulci loop, interior loop, junction loop, exterior loop, and so on. Okay. So this is the number of secondary structural elements involved in LNCRN-mRNA binding interface from RNA subserver. We use RNA to find the binding interface between each LNCRN and mRNA. So in this interface, we find that PBT1, only 4 helices of PBT1 are involved in interaction with DNA chassis, while in interaction with TRMT1, 3 helices are involved. And 3 helices are also involved with RNPF1 interactions. And so this work is continuing. the structural basis of action of LNCRNA through molecular talking and dynamics. We will do it further to understand it. We will also do experimental validation of predicted results by molecular biology experiments by a knockdown of LNCRNA of interest using siRNA technology and assessing the expression levels of targeted gene at mRNA protein level after knockdown by qRT-PCR RNA fission protein assay. So I think my PhD student, former PhD student, Dr. A. Salim Pasha, who is now a postdoc at NIH USA, and two senior Tiber, she present PhD student working, and also all the past and present students here, working in different fields in my laboratory. Also acknowledge the funding given by Department of Biotechnology and UVSAP DRS1 to our department. Thank you. And by this, I'm able to take any questions.
Thank you so much, Seema. So there is a question posted in the chat that was curious about where the protein information came from. What was the source of the protein data?
Protein data is Human Protein Atlas, that is a database of immunohistochemistry data. And in it, immunohistochemistry data for all of the different cancer types as well as normal cells are present.
So I had a question about, you showed the anti-correlated long coding, or the anti-correlation between RNA and protein expression in some of those cases. And you're thinking that those are regulated by long coding, non-coding RNAs? Is that your underlying assumption, and that how much have you gone into actually studying those specific interactions? Are you thinking those are inhibitory directly of the long coding, long non-coding RNAs on the mRNA blocking protein expression? And then how would the opposite work? If you've got low RNA expression, how would the long non-coding RNAs increase protein expression?
This is a very good question. No, so I haven't had time to check into that, but we have just got a regulatory network construction on it. So if this LNCRNA is increasing or decreasing protein expression levels, we have to look into that data. Thank you.
What proportion of the total gene expression changes did that make up? It seemed like it was a pretty low number. So the anti-correlated protein and RNAs.
Jessica, can you please explain again this question?
What fraction of the RNA changes were anti-correlated with protein changes over the overall? So okay. How many, or what proportion?
Okay, so most of the genes were positively correlated with the, okay, in this slides, most of the genes were positively correlated, I know. Anti-correlated, negatively correlated genes were less in number here. More out of 34 out of 70 genes, 34 were positively correlated genes and less in number were negatively correlated.
We have a question in the chat box. They're asking, I know that some long coding or LNC RNAs have been characterized in EMT, but I think you said that these RNAs are related to metastasis, were not part of the results of your analysis. Do you have any thoughts on why that might be?
Actually, the Cancer Genome Atlas it has gene expression data for primary tumors only, and not for metastatic tumors. So what gene expression changes we are observing, it is for primary cancer. That is why we are not able to observe that for metastatic ones.
Thank you.
Am I making myself clear?
I think so, yeah. If anybody else has any questions, please feel free to drop them in the Q&A box.
So do you think, or do you have any evidence that these long non-coding RNAs are directly interacting with proteins as well? I mean, there's obvious precedence with ribosomal RNAs. Do you think the LNC RNAs are also able to regulate protein expression in different ways?
Okay. So my student had made protein interaction network also, which is not presented here. And I remember that same case was in the protein interaction network also. So it may be regulating at protein level, although I have to find out for sure about that.
Right, we have a question in the chat from Dora Abodo. Has there been any work to look at translating some of these pan-cancer targets into drug trials?
Yes, not as of now, but we are looking towards it. Actually, E2F1 and ETS1, these are very good drug target that some people, some groups are working on this one. So we are quite sure there are some drugs available for these one. So I have not any idea.
Thank you.
Not validated.
All right, if there are no further questions, maybe we can move on to the discussion portion. Darren, what do you think?
Okay.
I think that's a good idea. Do you have the link handy?
I will
pull it up. If not,
chat.
we'll place it in the chat. So yeah, the discussion portion is handled on with a different Zoom link so that we can be directly interactive. The webinar format makes it much harder to have the direct interaction. So we will be switching over to the more interactive Zoom format, and Kavya will be putting the link in the chat. So we will start up over there. We'll give the speaker a couple minutes. So we'll start up there at 10:10. We'll go over there, but we'll get actually going at about 10:10. So we'll try to make things available as soon as possible. All right, thank you.
Thank you.
Thank you so much.